Hello, everybody. Can you hear us? Yes? So we will start because we're already 7 minutes late, so we have to start with the first, who is not here. Yes. So the first speaker of this session is Yu Martin Luong. He did his PhD in France, in Nantes, and now he's working at ANSES in Maison Alfort, near Paris. And he will talk about a model to predict SARS-CoV contamination of surfaces and mid-cuts in processing plants. You?
Good morning, everyone. Thank you, the Food Micro organizer, for having me today. My name is Yu Martin Leung. I'm from ANSES, French Agency for Food Safety. And today I will talk about some modeling approach, a nature-based model to predict SARS-CoV-2 contamination of surfaces and mid-cuts in food processing plant. So what's the problem between food and SARS-CoV-2? So few words about the context. As you may know already, since the beginning of the COVID-19 pandemic, several COVID-19 clusters have been identified in the food production sector. Since this sector was considered as essential sector in order mostly to keep running the food supply chain, to continue to provide food for people. Also, food was still produced during the pandemic, so if the food products are contaminated, hypothetically, the cluster could be expanded to more distant location. For example, as you see in this study here, the author detect the SARS-CoV-2 RNA copy on different sample of food, of raw food frozen across different cities of China. So one had the need to understand the transmission of SARS-CoV-2 in food processing plant for many objectives. Firstly, to protect the worker, mostly of this essential sector, from COVID-19 infections. And secondly, to avoid disrupting also the food supply chain in reason of the closure of the plant. So to predict the transmission of SARS-CoV-2, we developed in this project a mathematical to predict the transmission from preventing a cluster from happening. So to predict the transmission between workers in food processing plant and also the possible contamination of food products. I'm not going to any mathematical details today because it will take me 1 hour and a half. I will just give you some general principle of the model. So the type of model is called an agent-based model. So to describe different agents, different things within the meat processing plant. And the model is divided in different compartments. We have 4 here, different compartments, different modules to describe respectively the workers, the air, the food, and the surfaces in the meat processing plant. And the goal is to describe the interaction between these modules, and the principle is to know how the virus can be transmitted between models, for example, by respiratory activity of the workers, exhalation, inhalation from and to the air. And secondly, depending on the ventilation condition, the droplets containing the virus can fall on surfaces and also on food product. The same way by the workers. And at last, we can have also the transfer between the surfaces and food products if they are in touch. So the model has the different parameters. On one hand, we can have, we include several input data and assumptions to include mostly the characteristic of SARS-CoV-2 virus, for example, the variant of concern, the attack rate. And on the other hand, by doing some investigation, we would like to know how the model can behave under the different scenarios, depending on the different extrinsic factors like the dimensions of the plant, the ventilation condition, temperature, humidity, etc. And also, depending on the behaviors of the workers, are they wearing a mask? What are the activity in the plant and also outside the plant? And we can use the model to do different simulation, giving us different outputs to predict the infections of the worker, the food contamination, and also the surfaces contamination. And with these outputs, we can compare with all the data, all the epidemiological data for perspective, for model validation perspective. So how do we model the meat processing plant? So we implement the model in the way that the user can put in input of the model the different characteristics of the plant. As you can see here, the dimensions, for example, the different spaces. You can see the big area, which is the cutting room, and then some spaces, some classic space in the plant, an office, an entry hall, a waste area, a restroom, a cooling area for food storage. And we can put also some object in the plant, for example, the conveyor to transport food products from one point to another, some equipment, for example, for packaging machine. And once we have the plant, we can create the different workers in the plant. And in this model, we include different type of workers in the plant. Firstly, we can have the cutters. So the cutter who will stay mostly around the conveyor, about the table. And secondly, we can have the logistic worker who will transport the initial food products from the entry to the cutting tables. And finally, other logistic worker to transport the final product into the cooling area for storage, for example. And and also we can have other workers like a manager, administrative. We stay mostly in the office, sometime in the cutting room. And at the end of the day, we also have the worker from the cleaning team. And once we have the worker, we can generate this time the processing of meat products during the working day. As you can see here, we can have the unprocessed carcasses as the arrival gate, so the big carcass here, or the big half carcass. We go through different cutting steps, so they become smaller and smaller. And at final, they will put in the equipment package, machine, and the cooling area for storage. And when we have all of these things of the agent, we give different rules for every agent, including the workers, the services, the meat cut, to track, in order to track every position and every properties of every agent at different time point of the day and even after several days. And how about the virus? Firstly, we can have the infectious workers. So keep in mind that in this model, we model different state of the infection from the new infection until the recovery phase, including the period when people get some symptoms. And it appears that people are most infectious when they develop symptoms or not, including also a few days before they actually feel unwell, before they actually get sick. So we include all of this duration in the model. And we have some infectious worker, and depending on if they wear a mask or not, they will spread more or less a quantity of droplets in the air through the different activity like coughing, sneezing, or talking. And the droplets can be various size, and depending on the ventilation system, the ventilation conditions, the droplets can fall onto on different surfaces of food product. And the bigger ones may fall directly from the workers to the surfaces. And we don't forget that we can also spread the droplet onto other people if they are physically close. So for another worker, depending on the quantity of the droplets inhaled by this worker, they will have a risk to get infected. So we can have a contamination inside the plant through the air. We also model the transfer between the transfer of the droplet between the surfaces and food portion. And if we take the balances of the at the end of the day, we can have also the contamination of surfaces and meat cut. And something else, I say outside of the plant, we also take into account all the possible contamination sources of COVID, depending on the prevalence, on the epidemic situation, and also by other community activities outside of the plant. For example, if one live with some infectious at the same house, for example. Today I will show you some scenario simulation. So I will just begin with one simple scenario. We create a plant with a dimension like this, 21 by 26 meters, with 2 conveyors in the plant. We create 100 workers with 1 initial infection. We suppose that 80% of the workers wearing a mask. 67% will develop the symptoms, and if they are symptomatic, they will have a very high chance to be absent, so to stay home. We run the model with a time step of 5 minutes during 42 days. The model will implement under air. We also do a convergence analysis to take into variability, and all of this was done online. So some results. So the firstly, for the worker infection, I will show you here, for example, the evolution of the number of infected workers over time during 42 days. We can have different curves. The first one is the cumulative number of infections over time at the end of the period, sorry. The daily number of infectious workers, including the symptomatic, the asymptomatic, and also the pre-symptomatic workers, the person with non-infectious status, and we can also see in this graph the different sources of the contamination, for example, by the air, by the epidemic situation, and the community activities. Keep in mind that this graph on the left side is just one illustration of one simulation. So we had to do several simulations to take into account for the same scenario, to take into account the variability. So sometimes we can have the cluster or not. And here we can have different options to study different effects. For example, by changing the parameters values to study the effects contamination. So very quickly, I will show you the first effect, for example, the effect of social distancing. And how we did it is we just changed the length of the cutting table of the conveyors to change the distance between workers. So I will show you here the surfaces contamination. So we will just calculate the proportion of the surfaces with more than 5 log RNA copies of viruses. And as you can see here, if we increase the length of the conveyors, meaning that we increase the distance between workers from 1 meter to 2 meters, we can have, in average, a reduction of the surfaces contamination from 10 to 5 to less than 5%. The same thing for food contamination. If we look at the proportion of contamination, we have also a slight reduction of daily food contamination if we increase the distance between workers. And the last one is another indicator rather for the economic aspect. So sometimes workers are symptomatic and they may have been absent, so we can have a loss of production. And due to this absence, we can have the change in the production loss due to the effect social distancing. Here we don't see a very clear influence of production capacity. But if we look at the second effect of that I want to show you today, effect of mask wearing, if we change the proportion of workers wearing a mask from 0 to 100%, as you can see here, we can see a clear effectiveness of mask wearing to reduce surfaces and food contamination, and to maintain very efficiency, the production capacity, and so to avoid food processing disruption. So just to conclude on my presentation today, we developed a nature-based model with the possibility to change different parameters as input to define the different scenarios. This model can provide the different output to predict at the same time the contamination of the workers, the surfaces, and food products contaminated by SARS-CoV-2 virus. And this model takes into account several sources of variability based on prior knowledge and or data. Some work in progress, mostly the sensitivity analysis to deal with parameters uncertainties. The simulations can be compared with other data for validation purposes. And we can also have other potential applications, for example, prediction of SARS-CoV-2 contamination on food products depending on the different prevalence. And for example, for the surfaces contamination, we can also identify the most probably contaminated spot in the plant, maybe to improve hygiene. I would like to thank at the end the French National Research Agency for the funding of this SARS project, aiming to describe in a very global scale the transmission of SARS-CoV in food processing plant. You can see more details about this project SARS behind this QR code, and we have also a poster. We'd like to thank also all collaborators from the SARS project and also the technical support for members of Migan platform for calculation online. And thank you for your listening.
Thank you, Hugh. Are there any questions? We have time for 1 or 2 questions. We don't see any question on the platform. There is one question here.
Hello. So thank you for the presentation. Do you also took into account like the vaccination, or do you think it can be added to the model?
We talk about. We already talked about it. As I said, we consider the different infection state from newly infected, pre-symptomatic, etc. And the last state I didn't show you here is vaccinated. So maybe it doesn't play a role in simulation with a period of 42 days, but maybe it can play a role when we have a longer period of simulations. For example, for recontamination after some duration, for example, and maybe the attack rate also can be more or less sensitivity to the state of vaccination.
Okay,
but we talk about it.
And since we are kind of finishing, let's say, but it's always risky to say that with COVID, do you think how feasible it is, or how easy it will be to adapt your model to hopefully not new epidemics or new viruses that has different transmission patterns, and that maybe you have to adapt your model to this new data? Is it feasible, or is it the point of this model to be used in future epidemic?
Yeah, we were in the project was very focusing on SARS-CoV-2 because it was emergency, but we thought about it to maybe in future to extrapolate for the first time for the other respiratory viruses. And as you can see in the model, we can have different modules, so we can turn it off or turn it on. Some modules that we don't need for maybe for other pathogens. For example, we think about food pathogen, pathogen in food, Listeria or other. So for Listeria, for example, we will not have the contamination from the worker, but maybe from the surfaces. So the initial point of the model will not be the same, but there will be a possibility to extrapolate to different conditions, and maybe why not different epidemic situation.
Okay, thank you.
Right, thank you very much. Very interesting, very, very hot topics. Our next talk is by Tande Paksa from the University of Debrecen, working in the team of none less than Professor Joseph Barani. and she's going to give us a talk about the Milky Base database. Kudos on choosing the name, by the way. It sounds like a chocolate brand.
Good afternoon. My name is Tünde Paca. I am the PhD student of the Doctoral School of Nutrition and Food Sciences of the University of Debrecen from Hungary, where the chairman is mentioned earlier. I am a member of the Predictive Nutrition Research Group. In this presentation, I will describe the development of the Milky Base, the database of the biochemical composition of the human milk derived from the scientific publication. I will also highlight the novelties and the possibilities of this database, as our aim is to help researchers with resource and ontology for collecting their data in this format, in this common format, on the other foods component. Here in Athens, I would like to start with the words of the famous Greek physician. Hippocrates, the father of the Western medicine, said, "Let the food be thy medicine, and the medicine be thy food." Although the impact of the diet on our health is recognized more than 2,000 years ago, the nutritional databases still do not list more than 99% of the molecules that can be found in the food. As Albert László Barabási, our collaborator and one of the originator of the idea, called it in his publication, the dark matter of nutrition. We know it's there, and that's it. If we want better understand the impact of the diet on our health, we need a well-organized database which can be described this complexity on molecular level and any food components, where both published and experimental data can be recorded. The human milk, our first and most complex food, was the perfect starting point for our project. Its health benefits are proven, and the composition of the human milk is dynamically changing during the breastfeeding. So this gave us the idea to consider the human milk composition as a response for the environment and the history of mother and child, like a microbial response to the food environment. And this idea is not so far from the idea which is behind the COMBASE. Many of yours know this is a food microbiology database. In our data collection process, we first defined the ontology. For a start, we searched the relevant publications on human milk composition. Using FoodMine, a natural language processing algorithm, we got a list of papers which was refined and extended by human manual methods. The collected papers were read and scored by us human reviewers. The main selection criteria was that the publication should contain quantitative data on human milk components, what's more, so-called dynamic data showing the variation of the human milk during the time. Sorry. The next step, finally, we would like to manually evaluate all of the papers that we find and search for the human males composition, which are responding to environment. Mainly, we would like to find the data which is collected within tables, because it's helping us system to input our data to the Excel format, which was chosen to store our data. After define the ontology, during the populating the database, we continuously validating and curated the data which was added to the database. It was, one hand, it was made by syntax check, which was held by VBA macros developed by us. On the other hand, the semantic validation was performed to identify the anomalies within the publications. The latter one is much more difficult because it needs human intelligence to find these anomalies. After describing the data collection process, I would like to show the three main novelties of our ontology. The condition and the component fields are playing the role of the explanatory and the response variables modeling. Both static and dynamic data can be recorded together with their quantified uncertainties. And the general data structure, which were both the condition and the components fields, can be nested in this tree structure. As I mentioned earlier, the human milk is a unique, complex biological system where the components are continuously interacting with each other. These interactions are influenced by many factors, so-called conditions here, like mother's diet, milk stage, geographical location. The main novelty of the Milky Base is that it focuses both on the conditions under the data was generated and the effects of these conditions on the human milk composition. As you can see from these examples, which is highlighted from our database, all of these data should be recorded within just record. The first table shows the explanatory variables, which is the mother's diet, and the second table shows the response variables, which is the concentrations of the alpha tocopherol and gamma tocopherol within the human milk. Every new record, it is mandatory to enter the source of the information, the region, the size of the cohort, the used measurement methods, and at least one condition and at least one component field. This can be extended numerical values, you can see here, with their uncertainties, and all of this can be so-called dynamic values, which showing the data variation within the time. And the latter one can be plotted, the built-in macros. The next novelty is that both the explanatory and response variables are included in the tree structure, which is the general data structure of the database, which means the entries should be organized into a hierarchical classes. You can do this direct and indirect way, i.e. you can specify the ratio of the molecule in the group or directly the concentration. That means you can analyze not only the concentration of the molecule, but the group which is including the molecule can be analyzed. In this example here, the specific fatty acid, the arachidonic acid, showing the leaves of the tree. The including groups, the fatty acids and the lipids, are the branches of the tree, and itself the human milk is the root of the tree. The milky base offers a wide range of uses. Via the attached macros, the users obtains resources to generate plots, find patterns in dynamics, in dynamics on the human milk, and its dependence on the composition and the conditions, quantify and rank the importance of the conditions, and you can make predictions or make decisions. To conclude my presentation, I would like to get back the thoughts I mentioned at the beginning. A well-structured database like Milky Base, combined with numerical tools, has a huge potential for exploring the complexity, not only the human milk, but any other food compositions, and find new ways for understanding the composition of our diet and its impact on health and disease. In addition to providing resources to analyze the components for the researchers, our aim is to provide a template to inputting their data in this uniform format. We collaborated with the Barabashi lab in the creation of the Milky Base database, and the related paper was accepted for publication in the journal Scientific Data of Springer Nature. The Milky Base database, the connecting VBA macros, which was mentioned earlier, the user manual can be, it is available from the Fiction repository. Our second paper is under review in the first special issue. And finally, I would like to thank for our collaborators, for my colleagues within the Predictive Nutrition Research Group, and especially for my supervisor, József Baranyi, whom helped and advised, without whom it wasn't possible, this project. Thank you for your attention, and I'm happy to answer your questions.
Thank you very much, Tony. I think we have time for one question from the audience. All right, thank you very much. So our next speaker is Dr. Carlos Sabater, who is going to give us a very interesting topic, at least from the title, Machine Learning Model to Design a Novel Microbiome Targeted Food. Carlos has joined us online now. I hope you can hear us and see us. Carlos?
I can hear you. Can you hear me?
Yes.
Yes.
Thank you for the introduction, and I would like to thank the organizers for giving me the opportunity to present the following work. In the first place, food waste management is a key issue to global food security and friendly environmental governance. Worldwide, 1/3 of food produced for human consumption is lost or wasted along the food supply chain, primary production and food processing, representing the most significant losses. In addition, there is an increasing body of evidence that points towards vegetable food waste as a rich source of a wide array of carbohydrate structures that may exert a potential prebiotic activity. In this sense, the health-promoting effects of prebiotic compounds are usually defined by the combination of three following factors. The first one, the stimulation of commensal and beneficial groups. Here in the slide, you can see some examples of these health-promoting genera like Bifidobacterium, Lactobacillus, Ruminococcus, or Lactobacillus. The generation of beneficial microbial metabolites, such as short-chain fatty acids, and the inhibition of potential pathogenic bacteria. Also, oligosaccharides from vegetable byproducts include a growing range of emergent and candidate prebiotic structures. Here you can see some examples like arabinosyl oligosaccharides, isomalto oligosaccharides, and also oligosaccharides derived from pectin. These vegetable oligosaccharides have complex structures involving different combinations of monomers, such as galacturonic acid, arabinose, and ribose. In the slide, you can see an schematic representation of some of these common structures. Prebiotic substrates might exert their effects on microbiomes comprising hundreds or thousands of different species and strains. In this regard, the development and recent advances in next-generation sequencing technologies allows the characterization and the study of this complex community. Also, machine learning algorithms, which involve pattern recognition models, have been developed to process metagenomic and sequencing data and could be also used to elucidate structure-activity relationships in the field of prebiotics. Recent studies in the carbohydrate fields reporting application of machine learning include the identification of patients with gastrointestinal diseases, the assessment of clinical effect of common prebiotics like GOS and FOS, and the simulation of the full colonic fermentation process for different prebiotic conformations. Therefore, the aim of this work was to develop a novel computational framework for the rational selection of potentially prebiotic vegetable byproducts. For this purpose, different machine learning algorithms and other computational tools were applied to publicly available data in order to establish structure functions relationships of novel vegetable oligosaccharide mixtures. Here in the slide, you can see the methodology used. In the first place, we perform a bibliographic search to collect up to 15 articles reporting carbohydrate composition in terms of monomer characterization of different vegetable byproducts, as well as fermentation data and beneficial bacterial growth of health-promoting species determined by culture-independent techniques like qPCR and 16S sequencing. Once we collect this data from the literature, we computed a series of machine learning models, such as a model combining an artificial neural network and principal component analysis in order to elucidate a characteristic pattern for each byproduct according to the bacterial genera promoted, the byproduct source, as well as the treatments used to obtain these byproduct fractions. Finally, we performed an additional study focused on carbohydrate metabolism. In this sense, we annotated a large number of different reference genomes from some of the bacteria that were stimulated by this substrate. And for this purpose, we use the carbohydrate active enzyme database. In this slide, you can see the most relevant results from the machine learning model. In this sense, the combination of artificial neural networks and PCA revealed the influence of vegetable source and structural features of oligosaccharides, resulting in different biological effects. For example, in vitro fermentation of pectin oligosaccharides derived from artichoke, bergamot, and sunflower byproducts showing high uronic acid in their compositions promoted the growth of Bacteroides, Enterococcus, and Prevotella. In addition, the in vitro fermentation of alpha-galactose oligosaccharides and silo oligosaccharides from barley are likely to promote atopodium, while Bergascov citrus byproducts showing high verbose contents may stimulate different Clostridium species. We also determined some statistical correlations between different genera, including Butyrivibrio, Citrobacter, Mogibacterium, and Oscillospira. It should be noted that these genera comprise emerging probiotic species. These genera were also associated to different types of carbohydrate structures, which were mainly peptide oligosaccharide structures. These associations could indicate synergistic metabolism of these genera in the presence of prebiotic structures derived from vegetable sources. Finally, with regard to the carbohydrate metabolism study, it should be noted that comparative genomics of gut bacteria revealed the presence of characteristic functional domains from pectin-degrading glycosidases in the reference genome from several emerging probiotics, including Enterococcus, Eubacterium, Calicibacterium, Lactospira, and Prevotella species. These sequences involve a wide range of activities associated with pectin metabolism, including polygalacturinases, rhamnosidases, and pectin liase. The main conclusions of this work were the integration of the body of knowledge within the field of vegetable food waste valorization allows the rational selection of substrates with promising prebiotic activities by exploring the interactions among dietary fiber and gut microbial ecosystem using computational tools paired with structural, function, and genomic data. We can identify substrates with the potential to selectively stimulate gut commensals. The present work establishes a novel framework for prebiotic design. This tool could be extended to a wider range of commensal microbes and carbohydrate structures involving other type or prebiotic families. The present work has been funded by the following project. And finally, I would like to thank you all for your kind attention, and I will answer any question you may have. Thank you.
Thank you, Carlos. We have time for questions, either from the audience or online.
Online there is nothing.
Okay.
But then I have a question. Maybe I didn't catch what you told, but when you were talking about annotation of the Kazim,
yes,
I think you told only it's a lot, but 210 genomes, and I was wondering, was it how many species and strains does it encompass, and also did you consider the plasmidic genes, and also the intraspecies variability?
Okay, thanks for the question. This study is a preliminary study. We only consider a limited range of emerging probiotics, approximately 20 different genera, and we selected the sequences, the reference sequences that show a higher degree of completeness in the European nucleotide database. However, at this moment, we are performing additional studies, including a wider range of genome and reference assemblies. up to 2,000 or 3,000. In this sense, these new studies could reflect in a more appropriate way this intra-species variability.
Okay, thank you very much. Nice job.
Thank you.
Right. Thanks again, Carlos. I don't believe we have further questions. So we're going to move to our next talk by Elena Tastlati. Elena is an epidemiologist at the National Veterinary Institute in Uppsala, and the talk is about the detection of Campylobacter in the swine bowel, the impact of selective media time between sampling and start analysis, and the number of colonies confirmed, potentially the winner of the longest title. Well done.
Thank you. And first, I would like to thank the organizers for this inspiring conference, and second, for giving me the opportunity to present some of our results. So, in fact, I'm talking about optimizing detection of Campylobacter species in porcine cecal context more than optimal. Yeah, on background, so Campylobacter are gram-negative microaerophilic enteric bacteria that cause gastrointestinal infection in humans. Campylobacteriosis is the most notified zoonosis in humans in Europe. Before pandemic, the annual approximate number of reported cases was 200,000, but the true number of cases is way much higher. And this infection causes a lot of costs to the society, so there are estimations of 2.4 billion euros of the cost to the society. If we think about the sources of Campylobacter species, they are in the animal reservoir, and Campylobacter can colonize various animal species and have been detected in wild and domestic animals, especially birds, poultry, pigeons, passerine birds, and mammals, cattle, sheep, pigs, dogs, deer, and also insects. And in humans, the infection is mainly foodborne, caused, and poultry meat is a major source, but other contaminated meat as well as contaminated water, unpasteurized milk, and environmental infection is also possible, and direct infection. And the most common species in humans are Campylobacter jejuni, which causes approximately maybe 80% of the cases. It varies between countries. And Campylobacter coli, fewer, but it's more often resistant to antimicrobials, and that's why it increases its importance. But then there are other species that can be associated either with human infection or then sometimes in animal infection. Campylobacter is compulsory to monitor in Europe according to Zoonosis Directive and according to the communicable diseases regulations, and according to microbiological criteria, there's a monitoring of chicken neck skin. And then antimicrobial resistance is monitored both in human case patients and then in chicken, turkey, pig, and cattle. And then it's Campylobacter jejuni and coli. And so the EU Reference Laboratory of Antimicrobial Resistance, together with EFSA, made a survey some 4 years ago on the procedures when testing and detecting Campylobacter among the national reference laboratories of food and animals. And it was clear that there were big variabilities between the NRLs on the samples collected, whether they were pooled, whether a direct plate detection method was used or enriched method, what kind of selective media were used, what was the time interval between sampling and start of analysis, how the species identification was done, how many colonies were confirmed. And as there were so many variabilities, it hampers the interpretation of the collated monitoring data. Therefore, the aims of this study were to first investigate the optimal time interval between sampling and start of laboratory analysis, and then second, to define a second selective media for detection of Campylobacter jejuni and C. coli, in addition to MCCDA. MCCDA is the compulsory one, and it's also according to the ISO standard. And then third, to assess the number of colonies to be confirmed from the selective media. According to the ISO standard, it has been up to 5, and then, but in order to detect 2 species, this needed to be assessed. So the outline of the study, it was made in 2 parts. The first part was done at the EURL Campylobacter in Sweden, Uppsala. And the second part was done together with NRLs in Spain and Italy in order to gain more profound knowledge, since as we have learned here, the microbiota varies, and the same aims were assessed here. In the first part, which was done only in Sweden, so 15 pigs from 1 slaughter batch at a time were sampled at 1 abattoir at 4 time points, and then 50 milliliters of cecal contents were collected and placed in a clean jar, and then transported to the laboratory within 2 hours. Since the transport time was so short, there was no cooling needed. And then the second part of the sampling, 20 pigs from a slaughter batch sampled at an abattoir in Italy, Spain, and Sweden were taken, and then they were transported cooled to the laboratory of the NRL Campylobacter in Teramo in Italy and Madrid in Spain, and then EURL Campylobacter within 72 hours. And the methods used in both these parts, direct plating was applied, and then 3 selective media were used. It was MCCDA, which was made from oxoids dehydrated media. Preston was made in-house, and Butler was made from oxoids dehydrated media. And all these selective media were made by 1 laboratory and in Uppsala, and then dispatched to the laboratories in Spain and Italy in order to minimize the differences. And in the first part, the incubation was done at 37 degrees, microaerophilically, since they are microaerophilic bacteria. And in the second part, like is stated in the ISO standard, at 41.5 degrees. And then up to 5 colonies were confirmed if they were suspect or typical colonies. And the confirmation was either done using mass spectrometric method Malditov or then PCR method. That was validated, the PCR method as well. And then when we did the statistical analysis, in the statistical analysis we were considering Jejuni and coli, since they were the target pathogens for this recommendation. So a positive sample was considered if Jejuni coli was detected in any of the 60 isolates from a given sample. So there was a maximum of 60 isolates per sample. And a negative sample was if no Jejuni or coli were detected among these 60 isolates per sample. And then the association between media type, storage time, number of selected colonies, and the probability of detection were tested using logistic regression, where a nested random intercept for repeated measures was included on each sample, and then for the effect of the country. And then we also did a pairwise comparison of the selective media, and all this data analysis was done using the statistical package of R. And then here's just a summary of the detection first. In first part, so Campylobacter, some kind of Campylobacter was detected in 59 of the 60 samples tested. And in the second part, Campylobacter was detected in 55 of the 60 samples tested. And in both parts, it was C. coli which dominated among the isolates, and it's well known that C. coli is often the dominant species in pig reservoir. This high temperature was more sensitive for detection of C. coli than the low temperature. And C. jejuni was more found, in fact, using the lower temperature. And then those Campylobacter that are not thermotolerant could be detected using the lower temperature, like Campylobacter foetus, higher intestinalis, and Lanianae. Okay, then here I would like to concentrate on the differences between these three media when looking at the probability of detection of C. jejuni. So we can see that MCCDA and Preston were slightly similar for detection of C. jejuni, whereas Butler, the probability of detecting C. jejuni using Butler was much lower. On the contrary, when trying to detect C. coli, Butler was most sensitive for detection of C. coli, and Preston was slightly less sensitive than MCCDA. So there's these two species are not behaving in the very same way. Then if we think about the temporal aspect of the sensitivity, so the sensitivity probability of detection for C. jejuni. So this model indicates that the longer storage time reduced the sensitivity of the test compared to 22 hours of storage. But it was in fact only the 96-hour storage that was significantly lower than 22-hour storage, whereas for C. coli, a shorter time was already statistically significant. And it is also known that Campylobacter is relatively sensitive to storage. And here is depicted the impact of the number of colonies. So picking 2 or more colonies from each plate for confirmation was associated with increased sensitivity when compared to picking only 1 colony. And in these figures, yes, I'm sure. So Butler is green, red line is Preston, and blue line is MCCDA. So the conclusions: the analysis should be initiated preferably within 72 hours, but at last within 96 hours from sampling, and the combination of MCCDA and Butler seems to be most optimal for detection of C. jejuni and C. coli, and at least 2 presumptive colonies need to be confirmed. And then I would like to acknowledge my colleagues at the URL and also my colleagues at the 2 NRLs. And if you have questions or comments, so thank you.
We have a question on the front row here.
Hi, thank you. Sofia Strubia from Oniris, France. I would like to know if you use any particular storage condition for the long time of 72 hours. I mean, aerobic, refrigerator.
In fact, I could have mentioned in this, here we didn't use any special conditions. They were stored in a refrigerator during this time period. So there was no microaerophilic storage.
Thank you.
Okay, thank you very much, Elena. So our next talk from my colleague, the co-chair, Dr. Monique Zogaraj, the research director at INRA, the French National Institute for Agriculture, Food and Environment. And the topic of the presentation is using lactic acid bacteria as a biopreservation for foie gras. We're getting closer to lunch.
Thank you. Thank you to the organizing committee. I proposed the post there, and they proposed me a talk, and then they proposed me to be a moderator. So thank you. It will be my last talk in English in an international meeting before I will get retired within a few months. That's why I came with slippers. So what about biopreservation of foie gras, the fatty liver? Foie gras, fatty liver, has been produced and consumed since more than 5,000 years. The first traces have been reported in the ancient Egypt. Then it was spread to the ancient Greece and the Roman Empire. And then by the Jewish community, it was spread also in Europe, mainly in France and Hungary. That's why nowadays France and Hungary are the main producers. In France, foie gras is recognized as being part of the protected cultural and gastronomic heritage, and in Hungary, goose liver has obtained the distinction Hungaricum. So EU produces 90% of the world's production, mainly from duck, and to a less part from goose. And foie gras, or fatty liver, is obtained by overfeeding animals, ducks or geese. And if you see here, yeah, France is the main producer, followed by Hungary, Bulgaria, and Spain to a lesser extent. In France, foie gras is sold more or less half of the production is sold as half-cooked. So it means the liver has been cooked at low temperature, and then it is usually sliced and repacked. So it means that bacterial contamination can occur. Bacteria can stay after the cold cooking, I would say, and bacteria can recontaminate after slicing. And so there is a risk of regrowth during the storage of foie gras. That's why, in particular, in industrial production of foie gras, some preservatives are used, such as nitrates. But as you may know, nitrates are controversial, as nitrosamine produced from nitrates and nitrites may induce colorectal cancer. So in France and in Europe, there is now a tendency to diminish or even suppress the use of nitrates. So the question was, is biopreservation an alternative for reducing nitrates and still ensuring the microbial quality of foie gras? Biopreservation, as described by Stiles in 1996, is defined as extended storage life and enhanced safety of food using the natural microflora, nowadays we would say the microbiota, and or their antibacterial products. Lactic acid bacteria have a major potential use in biopreservation. We know that lactic acid bacteria belong to the foie gras microbiota. Depending on the products, they can reach 10 to the 2 to 10 to the 6 CFUs per gram. But the problem is that they may also act as spoilers of foie gras, in particular by producing lactic acid. And the other spoilers of foie gras, the other known spoilers of foie gras are Clostridia, like Clostridium algidicarnis or Clostridium frigidicarnis. So the strategy we used was to collect lactic acid bacteria collection from the foie gras and foie gras production environments, test their innocuity, means no production of histamine and antibiotic resistance, well, antibiotic sensitivity actually, and then to test their antagonistic activity against both lactic acid bacteria and Clostridia. We also we preferred to isolate some lactic acid bacteria producing bacteriocins. A good mechanism of antagonistic activity for biopreservation is nutritional competition, but we don't know so much about first the microbiota present in foie gras, even less about the metabolism of bacteria growing on foie gras. So that would be very interesting, but very difficult to monitor. The second way is production of lactic acid or organic acids, but as I told you, it may spoil foie gras. Another mechanism is production of H2O2, but we were afraid it could be also spoiling. So we were looking for a bactericide production. So we started from a collection of 70 isolates. Bacteria were isolated, some were isolated 10 or 20 years ago from foie gras or production line of foie gras. We identified them through 16S DNA sequencing. So as you can see, most were belonging to Lactobacillus sakei or curvatus that have been described as potential spoilers of foie gras. But we got also some Carnobacteria, Vessela, different Lactobacillus ansulato, Leuconostoc, and one Lactococcus lactis. All those strains were tested for the production of histamine, and actually 53 did not produce histamine above 50 ppm, and only 17 were just at the limit of that level. So we continued with all of those strains. Then we tested antibiotic resistance. For that purpose, we used the EFSA recommendation. We tested 8 antibiotics, and we used the CLSI document for the protocol using microtiter plates. You have here one example of a strain for which the resistance is just below the threshold recommended by EFSA. In total, we tested the 70 strains, and we were surprised that only 19 were sensitive to all antibiotic tested. As I told you, all those strains were isolated from foie gras or the foie gras production, so they are non-domesticated strains. They are just natural strains. And among those resistant strains, we observed that Carnobacterium was the genus where almost all the strains were resistant to at least one antibiotic. So we were surprised by this, and we decided to check a bit what was the cause of this resistance. We were using genome sequences of Carnobacterium maltaromaticum, where putative resistance genes were identified. Then we designed oligonucleotides dedicated to Carnobacteriaceae, and we tested the presence of putative resistance genes in our Carnobacterium collection. You see here an example of a strain that was resistant to tetracycline, and we could find that TET-M was present. This strain was also resistant to ampicillin, beta-lactamase was present, and also aminoglycoside resistance genes were present, and this strain was resistant to aminoglycosides. To summarize, for most of the strains, we had a perfect match in between the antibiotic resistance we observed and the presence of resistant genes. In some cases, we had only a partial match. For example, here, the strain was resistant to ampicillin and aminoglycosides and sensitive to aminoglycosides, although the genes were present. And in another example, here we have resistance to chloramphenicol, but the gene we targeted was not present. So it was just a one-shot study, but we were surprised and disappointed by the fact that so many strains were resistant, and for more or less 50%, we could really assess which gene was responsible for this resistance. So from the 19 antibiotic-sensitive strains, we checked then the antagonistic activity. We targeted three I don't know why I put it for it. 3 lactic acid bacteria and 2 Clostridium strains. In the first attempt, we did it simply on plates, and then we analyzed what kind of mechanism it could be. So from the 19 strains, only 11 were active against the 3 lactic acid bacteria strains, and among those, only 3 were active against only 1 Clostridium strain. None was active against Clostridium algidicarnis. So then we tried to get more information about the mechanism of inhibition of those 3 strains. For that purpose, we used supernatant, just normal supernatant, treated, neutralized supernatant to eliminate the pH effect, or heat it to see if the molecule involved in antagonism was sensitive to temperature. We also treated by catalase to see if the mechanism was associated to H2O2 production, and also treated by proteinase K to see if the active compound was proteinaceous. When lactic acid bacteria were targeted, we did it by growing lactic acid bacteria in liquid medium, and for Clostridia we did it on plates. So here you have two examples. Here you see we grow the target strain for 7 hours, and then we added the supernatant, either non-treated or pH adjusted or treated by catalase and so on. If you see here, the strain was active against, that was a Lactobacillus sakii, the target. After neutralization, the effect is a bit diminished, but just a bit. But after treatment with catalase, the effect totally disappears. So we think in that case it was the antagonistic effect was simply due to H2O2 production, maybe a bit lactic acid. And in that case, you see that the strain was active against the target strain, but as soon as we neutralize the pH of the supernatant, we have no activity anymore. So at the end, finally, from our 3 putative protective strains, none were active against Clostridium algidicarnis. And when they were active against the 3 lactic acid bacteria and against Frigidicarnis, it was through organic acid or H2O2 production. And I told you it was not what we were looking for. So actually, to conclude, the question was, is it possible to use biopreservation to ensure the safety of foie gras in the absence of nitrate? For the moment, I would say, well, at least we did not succeed. From the 70 strains that we tested, only 19 were antibiotic sensitive, 11 were active, but none produce bacteriocins. So what next? We have still strains in our collection, so we could go on with new strains. I would like to investigate the nature of the antibiotic resistance, because maybe some of the strains that are resistant actually don't have a transmissible resistant gene, so maybe we could go on with those. And I would like also to test them in foie gras, because maybe they could be active in foie gras. But the most important conclusion is that I told you already, more than 60% of our strains were resistant to some antibiotics. Those strains are just natural lactic acid bacteria. So I think we need a further investigation of those genes. Are they transferable? Are they plasmidic? Are they linked to transposons? And my message would be that most of the studies focusing on antibiotic resistance, they focus on pathogens. Okay, this is normal. But lactic acid bacteria in food, they are really dominant. And if those are antibiotic resistance, they can transfer it to other bacteria, pathogen or not pathogen. So I think, well, my conclusion is that investigation of antibiotic resistance in positive bacteria is really a need for the future. And I thank you for your attention.
Thank you, Monique. Have time for a couple of quick questions, I think. Maybe I'll start with a question regarding the antimicrobial resistance, which is when you showed that on a molecular level, it seems like a perfect match. Do you think that is related to any practices happening on the animal, on the farm level?
It is not supposed to. In France, but in many countries, antibiotics are forbidden as growth promoters. But they are still allowed as therapeutics. And we couldn't have any information about the use of the producers.
Oh, fascinating. We have a question in the middle of the audience here.
Thank you for this nice talk. I have questions about bacteriocin production. So I'm not sure to well understood, but how do you check the availability of bacteriocin genes inside
Well,
the species or the strains?
you mean in the genome or simply
Yes,
by...
both.
So by experimentally, it's by elimination. If the effect disappear after neutralization, after catalyzed treatment, and so on, so then it could be a bacteriocin. And then some bacteriocins are well known. So in the genome, you can you have Bagel. There are several pipelines to find bacteriocin production in the genomes. And then you can also, well, use the supernatant and trying to purify the molecule, I would say.
And the species you try to use contains these bacteriocin genes?
No.
No.
Well,
No.
we have not sequenced the 70 genomes.
Yes.
If we have a potential candidate, then okay, we could sequence. Actually, what I didn't tell is that we had a positive control, a strain known to produce nisin,
Okay.
and the nisin gene is known. I think it's plasmidic, so it's easy then to identify. But we are not at that stage yet.
Okay. Is it possible that the species contains the genes, but these genes are not expressed because of the absence, the lack of precursors in foie gras?
Yeah, well, yeah, and bacteriocins are, when a strain is producing bacteriocin, anyway, those genes are regulated. So at one moment it's not produced, but then it can be produced because there is an effector or something like this, the level of the population and so on. And you can have also mutated genes, for example.
Okay. So if genes are present, perhaps there is a lever in optimizing the expression of these genes.
Oh, yeah.
By adding, for example, some precursors or some prebiotics inside foie gras to enhance the growth and to enhance also the production of bacteriocin.
Prebiotics, I don't know. Why not? I don't know if anybody tried this. Very often it's the level of the population that is the signal for the bacteria to say, okay, we are too many here, I start to produce my bacteriocin. We always think from our human point of view, but we should think to the bacterium point of view. Why is bacterium producing bacteriocin? Not to help us to preserve foie gras or butter or whatever, simply to protect themselves.
Yes, you're right. Thank you very much.
Thank you, Monique, and please join me for another round of applause for her last talk in English. Well done. An achievement. Our next and final talk for this session is by Dr. Agni Hadjiluku, a lecturer in microbiology at the European University of Cyprus and also head of bioengineering in MBU. And the talk is delivered online. I can see Andrew joining us now about cell biosensors to detect listeria in dairy products.
Hello, can you hear me?
Yeah, nice and clear.
Okay, great. Initially, I would like to thank the organizers for this opportunity to present our research orally. I will present you a newly developed system for Listeria monocytogenes detection in dairy products based on a bioelectric cell biosensor. Aim of our study was to develop a biosensor system that could detect rapidly and robustly the pathogenic bacterium Listeria monocytogenes in dairy products. We chose Listeria monocytogenes as it's one of the greatest concerns in the dairy industry due to its ability to grow at very low temperatures, at the refrigeration temperatures, and due to the fact that listeriosis is a relatively rare disease with, however, very high hospitalization and mortality rates. For this reason, we developed the method testing the milk and the halloumi cheese samples. The newly developed system uses a cell-based biosensor technology and a portable device. The cell-based biosensor technology measures the changes in the cell membrane potential according to the principle of BERA, bioelectric recognition assay. The portable device is a multi-channel device with a replaceable connector of 8 screen-printed electrodes that has been developed by Embryo Diagnostics, a biotechnology company in Cyprus. And it's a device that can measure electric signals from biorecognition elements. The system is able to connect via Bluetooth with an Android device, and the results of the test appear on the screen, thus enabling the end user to be instantly informed of the test result. The creation of the biosensor, the cell-based biosensor, was performed by electroinsertion of the anti-Listeria monocytogenes P60 protein antibody into the membrane of mammalian cells, Vero cells. Due to the fact that Listeria monocytogenes may be present in food samples at very low numbers and usually accompanied by greater numbers of other microorganisms, 3 different protocols were evaluated based on the applied level of the enrichment broth. So the no enrichment protocol, primary enrichment protocol, and secondary enrichment protocols were tested and evaluated. All 3 protocols were based on the ISO standard for enumeration and detection of Listeria monocytogenes. More accurately, the first protocol, the no enrichment protocol, in this protocol, the food samples were not, in the food samples, there was no application of enrichment broth. The food samples were homogenized with sterile saline, and distant analysis was conducted. For the second protocol, the primary enrichment protocol, the food samples were homogenized with half razer, a primary enrichment broth suggested by ISO. And after incubation at 30 degrees for 24 hours, analysis on the samples was conducted. While for the third protocol, the secondary enrichment protocol, 100 microliters of the incubated food sample from the primary enrichment protocol were added in 10 ml of freezer, and after a 24-hour incubation, analysis was conducted on the samples. For all samples, for all protocols, both positive and negative samples for the presence of the pathogens were tested. The data obtained from the measurements were processed, and feature values were extracted and used for the algorithm development and the discrimination of the samples as positive or negative. Subsequently, thresholds were set for each feature value, and the limit of detection was calculated for each protocol and for both food substrates. Finally, the data by the measurements were compared, and the statistical differences were assessed through one-way ANOVA. Except for the analysis of the samples with the biosensor, the newly developed method, samples were also tested with ISO methods and molecular methods, specific PCR in parallel, and the results obtained from the biosensor method and the standardized methods were compared in order to calculate the performance synthesis of the newly developed method and the accuracy of the newly developed method. More accurately, we calculated the sensitivity of the method, specificity, the positive predictive value, and the negative predictive value. Our results. Our results from the no enrichment protocol indicated that the newly developed system had good discrimination power, discriminating negative from positive samples, and that the biosensor was not able to detect the presence of the pathogen, even if the pathogen was at very high population levels. This was attributed to food matrix effect. Also, the performance characteristics of the newly developed method were very poor for both food substrates. This was also the case when using the secondary enrichment protocol. The biosensor system was not able to discriminate positive from negative samples, but this was attributed to the enrichment matrix effect, since we were observing the same biosensor response in milk and halloumi samples. However, the primary enrichment protocol indicated very good results. We showed that the system was able to discriminate positive from negative samples in both milk and halloumi cheese samples with very high accuracy, having very high performance indices and with very low limit of detection for the pathogen. So to conclude, the procedure of the newly developed method was finalized as shown here. Initially, someone has to make a sample preparation based on the primary enrichment protocol followed. So the incubation that needs to be followed is for 24 hours. And after this incubation, the sample is added on the electrodes, the biosensor are added on the sample, and the device starts the measurement for 3 minutes. After the 3-minute measurement, the results appear on the monitor of the Android device and or in the online platform that we have created. Also the results from current and past tests can be retrieved and reported from the Belt Web platform. So we managed to develop a method that is very simple, that requires only 24 hours and that can detect the pathogen in milk samples and halloumi cheese samples at very low population levels with very high accuracy. Thank you for your attention.
Thank you very much, Angie, and spot on with the timing. So we have time for a question from the audience. So.
Hello, congratulations for your work. I wanted to ask, is there any advantage of the method you developed, considering that you need this enrichment step of 24 hours, if you compare this method with a qPCR method, for example, after this primary enrichment? Which is better, and is there any advantage?
The advantage is on the cost. It's a very cost efficiency method. We wanted to develop something that it's as quick as the qPCR, the PCR, and its cost is lower as the ISO methods. So I think we managed to do it. It's cost effective, and it does not require, of course, trained personnel to do it.
So then I have a question to complement this. Yeah, it's cost effective. It can be easy to use. Do you imagine to patent this method, to protect it?
Yes, we are now in this procedure to validate the method, to have a certification, and to protect our method, the newly developed method.
Okay, thank you.
I don't see any other raised hands, so yeah, perhaps you're just standing between the talk and the lunch. So thank you very much, Angie.
Thank you very much.